hello there. We didn't see you.
We were just talking about this news article and it really got us thinking about genetics.
Oh yeah, the world of genetics is wonderful. Let us illustrate just how interesting it is to this up-and-coming technology we were talking about.
Oh, I didn't hear you come in. We were just discussing human-animal chimeras.
This article talks about how some scientists are combating the shortage of organ donors by looking at using animals to grow human organs. In Japan, guidelines were altered so that animal-human embryos can be implanted in a surrogate. Previous to this regulation change, scientists had experimented with chimera embryos, but terminated the experiment a few weeks later. Pigs, rats, and even primates have been used as hosts for human organs. The bioethical concern is of cells migrating to non-target organs, especially the brain, resulting in animals with partially human cognition. Although scientists are proceeding with extreme caution, the idea of creating animal-human hybrids for the purpose of growing and harvesting human organs has many people concerned.
There's two ways to make a chimera. One is mentioned in the article by fusing two species, such as human stem cells, which then donated, goes into a pig embryo. And it comes back, and you get a developed kidney or a pancreas that is then donated back to the human.
The other type is naturally occurring. This type of chimera results when the utero of one embryo absorbs another embryo. So like these two pigs becoming one. This leads to the embryo gaining traits of the other that tend to have like different hair types, two different color dyes, or strange coat patterns, like this pig.
How fascinating! Let's dive into this topic more in depth.
Cell differentiation is the process where a cell changes from one cell type to another and occurs as different genes are switched on and off. For example, embryonic stem cell changing to a blood or liver cell. Cell differentiation allows adult somatic cells, for example blood cells, to be reprogrammed into pluripotent stem cells or iPS cells. iPS cells are undifferentiated cells reprogrammed to an embryonic-like state, which allows scientists to differentiate these cells into any cell type of the body. Induced pluripotent stem cells or iPS cells are generated by reprogramming, for example, adult blood cells. iPS cells are typically derived by introducing products of specific sets of pluripotency-associated genes or reprogramming factors into a given cell type. The reprogramming factors used are transcription factors, and there are typically 4 types. These factors then trigger a gradual process of silencing markers of the differentiated phenotype and inducing markers of the pluripotent state in some cells. As pluripotent cells, iPS cells have the ability to generate all cell types found in the body.
Fun fact, we could all have our own pluripotent stem cell line.
There are 2 forms of mRNA processing that are involved in iPS cell expression: RNA interference and methylation. Genes can be silenced through the process of RNA interference. This is when small strands of RNA molecules, either microRNA, miRNAs, or small interfering RNAs, siRNAs, are created from the cutting of double-stranded RNA by the enzyme dicer. They then pair with proteins to form an RNA-induced silencing complex, or RISC. These RISC complexes then pair with part of the mRNA and affect it in a way that results in gene silencing. siRNA degrades mRNA before it can be translated, and miRNA incorrectly pairs with mRNA and inhibits translation.
The second form of mRNA processing is methylation of DNA. DNA methylation refers to the addition of methyl groups to the nucleotide bases. The addition of these methyl groups can activate or deactivate different genes and their expressions. Because of these changes can be passed to future generations, DNA methylation is maintained throughout DNA replication. Methylation is of C bases only. And while methylation is passed between different generations of daughter cells, we do not know if it is passed through generations in animals. However, it does in plants.
This study just touches on the surface of this topic.
Yeah, no kidding. There's much controversy surrounding this topic. It will be interesting to see how it evolves in the future.
Crossing over metaphase, I can recombine. Take a look, it's a GMO, a genetic sequence.